Nearly 1.2 million people in the United States are living with HIV, and approximately 13% of them don't know it.
Hello and welcome to the HIVRNATestguide.com YouTube channel, your online partner for quick, affordable, and 100% confidential HIV testing. Whether you're looking for quick, confidential results or simply want to stay informed about the latest in HIV research, you're in the right place. For more information, visit HIVRNATestguide.com. Check the link in the description below or visit the bio section of our channel. You can access our more than 4,500 HIV testing labs across the United States, just a click away. So, without further delay, let's start today's video.
Hello and welcome to the HIV RNA Test Guide podcast.
Hello, everyone.
We are your trusted source for HIV testing information, and we connect you with a huge network of over 4,500 testing labs all across the United States.
That's right.
So whether you're tuning in to get ready for a doctor's visit, or maybe you're just checking up on the latest science, or, just taking charge of your health, we are really glad you're here with us.
We really are. And I think for today, we should probably set a specific tone for our time together.
Okay.
The information we're about to get into is, well, it's pretty significant. And it involves some fairly complex biology.
Right.
So our goal here is to be really clear and deliberate, accessible. We're going to speak slowly, take our time, and just make sure these concepts are fully understood.
No rush at all.
No rush. Just a commitment to really understanding what's going on here.
Absolutely. We're going to guide you through this, step by step.
Yeah.
So I want you to imagine the date for a second. It's February 2026. The world of medical research is always moving. But right now, the landscape of HIV research, it's shifting in a way that feels, different.
It's a very exciting moment. It really is. We're seeing this kind of convergence of technologies that's opening doors we've been pounding on for decades.
Right?
It feels like the fog is finally lifting on some of the most stubborn problems in immunology.
And the main topic for our deep dive today is one of those technologies. It has a name that sounds like it belongs more in an art studio than a high-tech lab. You're talking about DNA origami.
DNA origami. It does sound delicate, doesn't it?
It really does.
It makes you think of like paper cranes or little folded boats. But, make no mistake, this is absolutely cutting-edge science.
So not an art project.
Not at all. It represents a fundamental, a real rethinking of how we build medical tools right at the molecular level.
And we're going to be discussing a new study that was just published February 5th in the journal Science.
A major journal.
A huge one. And this study is hinting at what could be a functional key for an effective HIV vaccine. That's a big claim, and we're here to see if it actually holds up.
Right. So our mission for this deep dive is to understand exactly how folded DNA, how it could solve a problem that has baffled scientists for 40 years.
40 years.
First, a whole generation of researchers. So we're going to look at the mechanics, the biology, and, the implications. We want to understand the why and the how, not just the headline.
And to do that, we are drawing from some great sources, specifically excerpts from the paper itself, DNA Origami, a new frontier in HIV vaccine design, and also interviews with experts from MIT, Weill Cornell Medicine, and the University of Melbourne.
So we've got the best minds in the field weighing in.
We do.
So let's begin.
Okay, let's unpack this. Section 1 is, it's really about the challenge, right? To get why this DNA origami idea is such a massive deal, we first have to understand the problem it's trying to solve.
Absolutely. Context is everything.
So why is designing an HIV vaccine so ridiculously difficult? We have vaccines for so many other things, the flu, COVID, measles. What makes HIV so stubborn?
That is the million-dollar question. Actually, it's the multi-billion-dollar question, I guess.
Yeah.
And the core of the problem, really, is that HIV is a shifting target. It's a virus that constantly, and I mean constantly, reshapes its surface proteins.
When you say reshapes, what does that mean in practice for the immune system? I mean,
Well,
is it just putting on a new hat?
It's so much more dramatic than that. Imagine you have a lock that's constantly changing its shape.
Okay.
You have a key, and the key in this analogy is an antibody. And it fits the lock perfectly today. It slides in, it turns, it disables the mechanism. Great.
Problem solved.
For today. But tomorrow, that lock has morphed. The pins inside have moved. The cylinder is a different size. Your key, the one that worked yesterday, it no longer fits. It fails.
So the virus is essentially changing its disguise faster than our immune system can memorize what it looks like.
Exactly. But let's go a little deeper into that disguise, because it's even more clever than that. The virus doesn't just change shape, it covers itself in something we call a glycan shield.
A glycan shield. Glycan. That means sugar, right?
Yes. The virus literally coats itself in sugar molecules that it steals from the host's own body.
Wait. So it's camouflaging itself with our own biological material.
Precisely. The immune system is like a patrol, right? It's going through the body looking for foreign invaders.
Things that don't belong.
But when it bumps into an HIV particle, often the very first thing it sees is this dense forest of sugars that look exactly like the sugars on your own healthy cells.
So it just moves on.
It thinks, oh, this is just us, nothing to see here, and it moves on. It's incredibly devious.
That is evolutionary genius.
It really is. And then underneath that sugar shield, the actual protein spikes, the machinery the virus uses to infect our cells, those are the parts that are constantly mutating.
So it's got a disguise on top of a disguise.
You got it. Adam Wheatley, he's an immunologist at the University of Melbourne. He describes this design process as incredibly challenging. You're trying to hit a target that is hiding behind a shield and at the same time refusing to stand still.
So the standard antibodies that might work against one strain?
They fail completely against others, because by the time the body makes an antibody for the version it saw on Tuesday, the version circulating on Friday is already different.
That's a huge problem.
It's massive. And here's a statistic to put it in perspective. The diversity of HIV inside a single infected individual,
just one person,
can be greater than the diversity of the influenza virus across the entire globe during a flu season.
That is a staggering fact.
Wow.
It really puts into perspective why a simple flu shot approach just isn't going to work here.
Not even close.
But there is a solution, isn't there? Or at least a theoretical one that scientists are chasing.
Yes. Thankfully, yes. There is a holy grail of HIV immunity that researchers have been pursuing for decades. It's something called broadly neutralizing antibodies.
Broadly neutralizing antibodies. Okay, let's break that name down.
Yeah.
BNAbs, for short, I think.
BNAbs, yeah.
What does that mean in plain English?
It means that these are very special antibodies. They don't just target the parts of the virus that are always changing, the parts on the surface.
They ignore the distractions.
They ignore the distractions. They lock onto the parts of the virus that don't change from strain to strain.
Wait, there are parts that don't change. I thought you just said it was a shapeshifter.
It is, but even a shapeshifter has to have some consistent internal machinery to survive. Think about it this way. The virus has one job to do.
Infect a human cell.
Right. And to do that, it has to physically latch onto the cell and sort of pry it open. It needs a mechanism.
A key to get in the door.
Exactly. It needs a very specific key to open the door to our immune cells. And if the virus mutates that key too much, well, it won't fit the lock on the human cell anymore.
And if it can't get in the cell?
It can't reproduce. It dies. So there are these very specific regions on the virus that are conserved. They stay the same because they absolutely have to.
And these broadly neutralizing antibodies, these BNAbs, they target those specific unchanging spots.
Correct. They see past the sugar shield. They ignore the shifting disguises. They have this laser-like focus on the structural necessities of the virus.
Can you give us an example? Is there one of these we know about?
A prime example, and one that's really central to this research, is an antibody called VRC01.
VRC01. Okay, tell us about that one.
VRC01 was identified in a small number of people living with HIV. Their bodies, usually after years and years of fighting the infection, naturally figured out how to make it.
Wow.
And this antibody, it targets a very vulnerable region on HIV's outer envelope. It's called the CD4 binding site.
The CD4 binding site. That sounds technical. What is it?
You can think of the CD4 binding site as the actual tip of the key the virus uses to enter our immune cells.
The most important part of the key.
The most important part. The virus needs this site to get inside your cells. And because it's so crucial for the virus's function, it doesn't really differ much between strains. It has to be constant.
So VRC01 targets the virus's own key. It basically jams the lock.
That's a perfect way to put it. It binds to that site and physically blocks the virus from ever attaching to the human cell. And if the virus can't attach, it can't infect. End of story.
That sounds perfect. If we know this antibody exists and we know exactly what it does and we know it works, why don't we just give everyone a vaccine that makes the body produce VRC01? Why hasn't this been solved already?
And here's where it gets really interesting. and really, really difficult. We're now entering the realm of what immunologists call germline targeting and the problem of scarcity.
Scarcity. Scarcity of what?
The right cells. You see, you can't just inject the VRC01 antibody itself and call that a vaccine.
Right, a vaccine has to teach your body to make its own.
It has to train your B cells.
The B cells are the antibody factories.
They're the factories, exactly. But here's the catch. Every B cell in your body starts with a unique genetic sequence. It's pre-programmed to make one and only one specific shape of antibody. In your body right now, you have billions of B cells, all with slightly different key shapes.
It's like a library of billions of different potential keys.
An immense library. But the biological hurdle is that the specific B cells, the ones that have the potential to evolve into factories for VRC01-like antibodies, are extraordinarily rare in the human body.
So the potential is there, but the initial workforce,
Oh.
the starting cells, there just aren't many of them.
They're there, but finding them is like finding a needle in a haystack. Actually, it's harder than that.
How so?
It's like finding a specific needle in a haystack that is actively hiding from you.
Hiding. Why are they hiding?
Because that target, the CD4 binding site, it's not on the surface of the virus. It's in a recessed pocket. It's deep inside the protein structure. Most B cell keys can't even reach it. And the few B cells that can reach it often have shapes that look a little bit like they might accidentally attack the body's own tissues. So the immune system, to be safe, naturally suppresses them. It keeps them quiet.
So in order to fight the virus, we have to fight against our body's own safety mechanisms.
Yes. Rezan Drabi, an immunologist at the University of Pennsylvania, he calls activating these elusive cells an engineering problem. It's not just about showing the body the virus, it's about finding that one specific cell in a crowd of millions and waking it up while telling all the others to stand down.
And that leads us directly to the problem with our current tools, the noise of traditional vaccines. This is really section 2 of our deep dives.
Right. To understand the elegance of the solution, we have to understand why the old way wasn't working well enough. We've been trying to wake up these rare cells for years, but we kept running into the same wall.
So how do conventional vaccines work in this context? What's the standard approach?
Conventionally, if we want to make a vaccine for a virus like HIV, we use either weakened viruses or, more commonly now, synthetic virus-like particles.
Virus-like particles.
Yeah, these are nanostructures that are engineered to mimic the size and the geometry of a real virus. They look like the enemy, but they can't actually infect you.
Okay, so it looks like a virus, but it's harmless. A decoy.
A decoy, exactly. But you have to build these decoys out of something. You can't just have the important part, the antigen, floating in space. You need a structure to hold it up.
A scaffold.
Yes, a scaffold. And traditionally, these virus-like particles are built on protein scaffolds.
A protein scaffold. Can you give me an analogy for that? What does that look like?
Sure. Imagine a mannequin in a store window.
Okay.
You want to show the shopper a very specific jacket. That jacket is the HIV antigen. It's the important part. It's what you want the immune system to recognize and buy. But you can't just throw the jacket on the floor. It would be a crumpled mess. So you put it on a mannequin, the protein scaffold, to hold it up, give it the right shape, and display it properly.
That makes perfect sense. You need the mannequin to give the jacket shape and make it visible. So what's the problem?
The problem, the conflict, arises because the immune system is very, very thorough. It doesn't just look at the jacket.
It looks at the mannequin.
It looks at the mannequin too. And in this case, the mannequin is made of a protein that the body recognizes as foreign, as not self.
So it attacks the mannequin.
Yes.
Yeah.
And it attacks it aggressively. The body launches what we call an off-target antibody response against the scaffold itself. And because the scaffold is usually big and obvious and easy to access, unlike that tiny recessed HIV binding site,
The immune system actually prefers attacking the easy target.
it prefers the mannequin over the jacket. We call this phenomenon immunodominance. The scaffold dominates the immune response. It screams much, much louder than the antigen we actually care about.
And that is the noise we keep talking about.
That's the noise.
Yeah.
Exactly. It creates immunological noise. It just dilutes the whole response. It's like you're trying to train a guard dog to recognize a specific intruder, but the dog keeps getting distracted by the mailman and the squirrel and the wind rustling the leaves.
Right.
The immune system gets busy fighting the scaffold, the squirrel, and it pays way less attention to the actual HIV antigen.
And since the B cells we need, the ones that are supposed to be targeting that tiny HIV pocket, are already so incredibly rare.
This competition makes it nearly impossible to reliably activate them. The noise just drowns out the very faint signal. The easy B cells that attack the scaffold crowd out the hard B cells that we need to attack the virus.
So logically, we need a way to present the jacket without the mannequin distracting the guard dog.
Yes.
We need a scaffold that is essentially invisible to the immune system.
That's a perfect way to put it. We need a delivery system that does not compete with its own payload. And that brings us right to section 3, the solution DNA origami.
Here we go. DNA origami. I still love this name. But we've established it is not paper art. So what is it, really?
It is pure molecular engineering. DNA origami is a precisely engineered three-dimensional scaffold that is made entirely of folded DNA.
Folded DNA. Now, I usually think of DNA as this long, stringy, double helix of code. How do you fold a strand into a complex, stable structure?
It relies on the most basic, fundamental property of DNA, base pairing. You know how A always binds to T and C always binds to G?
Yes, the letters of the genetic alphabet.
Right. So what scientists do is they take one very long single strand of DNA from a virus that doesn't infect humans. Then they mix it with hundreds of shorter strands called staple strands.
Staple strands.
And these little staple strands are custom designed in a computer to bind to very specific parts of that long strand. And by binding in all these specific places, they pull the long strand together, pinching it and folding it into a predetermined three-dimensional shape.
So it's almost like using hundreds of tiny programmed paper clips to fold a long piece of yarn into a specific shape, like a box or a sphere.
Exactly like that. And because we can program the DNA sequence of those staples, we can program the final shape with incredible precision. We can make spheres, cubes, triangles, almost anything we can design.
So they took out the noisy protein mannequin and replaced it with a DNA mannequin. Why is that better? What's the breakthrough?
This is the key. Researchers first tested this approach in an experimental COVID-19 vaccine, and they noticed something crucial. The immune system showed virtually no response to the DNA scaffold itself.
It was invisible.
It was a silent scaffold. The immune system just didn't see it as a threat. It completely ignored the DNA structure.
Why? Why does the body hate the foreign protein scaffold but just ignore the DNA one? Our bodies have DNA, but this is still foreign DNA, isn't it?
It is, but it comes down to how our bodies process different types of molecules. We have our own DNA floating around all the time from dying cells, and when DNA is folded up this tightly, it doesn't trigger the same stranger danger alarms that a big foreign viral protein does. It just flies under the radar.
So, going back to our analogy, the mannequin is now effectively invisible. The immune system literally only sees the jacket.
Exactly, and that's the whole point. Anna Romanoff, she's an author of the study from MIT, she noted that this property is especially useful for HIV. Because the target B cells are so rare, you cannot afford a single distraction. You need 100% of the immune system's attention on that one specific antigen. You need total focus.
It sounds like a complete game changer. But, we've heard good theories before. Did it actually work in practice?
They tested it in mice, and the comparison was very, very rigorous.
Walk us through the experiment. What did they do?
Okay, so they took one group of mice and they gave them the state-of-the-art protein nanoparticle vaccines, the noisy ones we've been talking about.
The current best in class.
Right. Then they took another group and gave them the DNA origami vaccine, the silent one. And here's the important part. Both vaccines were carrying the exact same HIV antigen. The only difference was the scaffold, the delivery truck.
Okay, so a perfectly controlled experiment. And what was the outcome?
First, the off-target responses. In the DNA group, they were sharply reduced. The antibodies against the scaffold itself basically vanished, just as they'd hoped.
The noise was gone.
The noise was gone. But the most important metric, of course, was the B cells.
The rare ones we need to activate.
Yes. And the result was that the DNA origami vaccine produced up to 3 times more of the important memory B cells compared to the protein nanoparticle vaccines.
3 times more. That's not a small improvement. That's a massive difference.
It is hugely significant. In the world of vaccine development, you are often fighting for a 10% or 20% improvement. A 300% improvement is just startling.
Wow.
Mark Bathe, he's a professor at MIT and one of the senior authors, he called this a potential breakthrough that could honestly transform vaccine design.
I bet even the researchers themselves were surprised by that number.
They were. Bathe admitted that even they were surprised that the DNA origami outperformed the standard particles so convincingly. They knew the theory was sound, But seeing the biology obey the theory that clearly and that strongly, that's rare.
It's just fascinating that simply removing the distraction, just turning off the noise, made the signal that much clearer. It really validates the whole idea that the noise was the core problem all along.
It does. John Moore from Weill Cornell, another expert in the field, he described the study as elegant. He said it clearly demonstrates that eliminating these scaffold-related immune responses, it pushes the immune response in the right direction.
In the right direction. That is exactly what we want. But I have to imagine you can't just fold some DNA, slap an antigen on it and call it a day. There must have been some engineering challenges. Science is never a straight line from a great idea to success.
Oh, absolutely not. And that's where Section 4 comes in. It's all about engineering the solution, because this was not an instant success. It's so important to understand that science is iterative. You try, you fail, you learn, you try again.
So what happened with the first attempts?
The early versions of the DNA origami vaccine, they actually produced really weak responses.
Really?
Yeah.
Why? If the scaffold was silent, shouldn't it have worked right away?
That's the logical question, right? But silence isn't enough if the package doesn't get delivered to the right address.
The delivery problem.
A major delivery problem. The early vaccines were failing to reach the specialized immune cells deep inside the lymph nodes.
The lymph nodes. That's the training ground for the immune system, isn't it?
It is. It's like the boot camp for B cells. That's where they hang out and learn what to attack. And specifically, they live in these little structures called follicles, which are deep inside the lymph node. If your vaccine can't get deep into those follicles, it can't train the B cells.
So the first DNA shapes were getting stuck at the front door of the boot camp.
Essentially, yes. They just weren't optimized for travel through the lymphatic system. They were getting cleared out too quickly.
So how did they fix it? What did they change?
They made it tunable. And this is the real beauty of DNA origami. Because you are engineering the structure literally at a molecular level, you can tweak it with incredible precision. It's like editing a digital file rather than trying to sculpt a new statue from stone.
So what did they tweak specifically?
First, they redesigned the entire structure to pack the HIV antigens more precisely and more tightly together. This change in the structure, just altering the spacing and the density of the antigens on the surface, that ensured that the particles were the right size and shape to be carried efficiently into the correct regions of the lymph nodes.
Okay, so that's better packaging for better delivery. That makes sense.
But then, once they solved that, they ran into a second problem. A really interesting problem that was, in a way, created by their own success.
Which was?
The problem of the silent scaffold itself.
Wait, hold on. I thought the silence was the good part. That was the whole point.
Right. It is good for avoiding distraction. But here's a really interesting nuance of immunology. B cells don't work alone. They're powerful, but they need permission to fully activate and fire. They need a second signal.
Permission from whom?
From another type of immune cell. From T cells.
T cells. Okay, we've talked a lot about B cells. What is the role of the T cell here?
Think of T cells as the helpers, or maybe the coaches. They provide support and encouragement to the B cells. They give the signal that allows the immune response to grow, to mature, and to become robust and long-lasting. If a B cell sees an antigen but it doesn't get a thumbs up from a T cell, it often just goes back to sleep.
Okay, so it needs that confirmation.
It needs that confirmation. Now, think back to the old protein scaffolds, the noisy ones.
Yeah, the ones the body hated.
Well, because the body hated them and saw them as so foreign and dangerous, it naturally recruited lots and lots of T cells to the scene to check out the commotion. The bad noise was actually providing the good T cell help purely by accident.
Oh, I see. So the loud, annoying alarm bells were a distraction, but at least they woke up the coaches.
Exactly. But because the new DNA scaffold is silent and invisible, it wasn't recruiting those helper T cells naturally. The B cells were finally seeing the antigen clearly, but there were no T cells around to give them the go-ahead.
That is an incredible bit of engineering irony. They stripped away all the bad noise, but then they realized they had accidentally stripped away a necessary signal too.
Precisely. So the scientists had to go back to the drawing board and manually add a helper molecule to their design. They engineered the DNA scaffold to also carry a specific known molecule that wakes up T cells directly.
So they engineered the silence to remove the distraction, and then they engineered a very specific, clean signal to bring back the help they needed.
That's it. They reconstructed the ideal immune response from the ground up, keeping only the parts they wanted and getting rid of all the parts they didn't. Adam Wheatley, the immunologist from Melbourne, he noted how efficiently they modified the scaffold. And he highlighted that the major utility of DNA origami is that it is really tunable. You can adjust the size, the shape, the antigen spacing, and now the helper molecules until you get the response exactly right.
That is the very definition of precision medicine.
Yeah.
So let's just walk through how this works inside the body now that it's been fully refined. What's the mechanism of this final successful version?
Okay. So imagine the journey. You receive the vaccine. The DNA scaffold enters your system. It's carrying that special antigen that mimics the CD4 binding site, the vulnerable spot on the HIV virus.
Right.
The scaffold then travels to lymph nodes. Because of its new optimized design, it drifts deep into the follicles where the B cells live. And because it's silent, the immune system doesn't waste any energy or resources attacking the scaffold itself. There is zero off-target noise.
The path is clear for the real signal.
The path is clear. So the vaccine can now effectively search for those incredibly rare B cells that just happen to have the right shaped receptors for the CD4 binding site. And because there is no competition from all those irrelevant B cells that would normally be attacking the scaffold,
It has a much better chance of finding them.
a much better chance. It can selectively bind to those rare B cells.
And the helper molecule you mentioned is there to call in the T cells.
Yes. The T cells arrive on cue, they give the thumbs up, and that specific B cell activates. It begins to divide, to multiply, and to mutate, constantly refining its ability to attack that CD4 binding site.
And that initiation, that's the on switch for generating those broadly neutralizing antibodies.
Yes, it's
Oh.
like using a sniper shot instead of a shotgun blast. It hits exactly what we needed to hit and nothing else.
That's a powerful image.
Yeah.
It really does highlight why accuracy matters so much more than just raw power in this particular fight. But, and this is the big but, we have to be realistic, this was a mouse study.
That is the critical question.
What does this mean for humans?
We must always, always be cautious when translating from mice to humans. As the saying goes, mice are not little people.
True. Their immune systems are similar in some ways, but very different in others.
Right. There are significant hurdles ahead. Reyes Andrabi, the immunologist, he stated it perfectly. He said, they have figured out the first step.
The first step, not the whole journey.
Not at all. HIV vaccination is just incredibly complex. It will likely not be a single shot. This is not going to be a one-and-done situation.
So you're thinking it would be more of a regimen, a process over time.
Almost certainly.
Yeah.
To develop a truly robust and lasting immune response, it might require multiple components, a series of different vaccinations to carefully guide the immune system over time.
How'd that work?
Well, you might need a primer shot like this one to just wake up those rare B cells, get them started, and then you might need booster shots with slightly different shapes or antigens to teach those B cells to focus even tighter, to become even more powerful.
So it's a multi-stage training program for the immune system.
Exactly. And John Moore also cautioned that we still need to see if this immune focusing, the silence effect, works the same way in humans. Our immune systems are much more complex and have much longer histories than lab mice.
Right. We've been exposed to more things. Our B-cell libraries are totally different.
Our libraries are different, so the silence might not be as perfect in us. It might not be as clean. We simply have to test it.
But the potential here seems to go beyond just HIV, doesn't it?
Oh, it absolutely does. This is what investors and scientists get very excited about. This is a platform technology.
Meaning you can use the same basic truck to deliver a bunch of different packages.
Exactly. If you can prove that DNA Origami works as a silent, tunable scaffold, you can then use it for all sorts of other things. You just swap out the HIV antigen for something else.
Like what? What are the other applications?
The study authors themselves suggest it could be applied to other rapidly mutating viruses, like influenza.
The flu. That makes perfect sense. We have to get a new flu shot every single year because the virus changes so fast.
Exactly. The flu has the same basic problem as HIV, just to a lesser degree. It has a head region that changes constantly and a stem region that stays pretty much the same.
So if we could use DNA origami to focus the immune system on that unchanging stem,
We could potentially have a universal flu vaccine, one shot that might last for years or even a lifetime.
wow.
Any virus where the vaccine effectiveness is limited by off-target distraction or by rapid mutation could potentially benefit from the incredible focus provided by DNA Origami.
It really feels like a whole new way of thinking about vaccine design. We're sort of moving away from the old idea that foreign is better, that you need a big, messy signal to stimulate the immune system,
Right.
and moving toward this idea of precision and silence.
That is the shift. Moving from these foreign protein scaffolds to silent DNA scaffolds. It represents a fundamental change in immunotherapies. It's all about control. For the first time, we aren't just, throwing things at the immune system and hoping for the best. We are engineering the interaction from the ground up.
So after all this, what does it all mean for you, the listener? We've covered a lot of ground today.
I think if we synthesize this entire deep dive, we've really journeyed from the core difficulty of HIV, the shifting target, and those incredibly rare B cells, all the way to the elegance of this new solution.
Right.
We've seen how scientists used a silent DNA scaffold to deliver a very specific message, but without all the background noise that has plagued every previous attempt.
It really is a story of persistence and just incredible ingenuity. And while we're obviously not at the finish line yet, this feels like a significant, tangible leap forward.
It does. It offers a genuine glimpse into the future of a functional vaccine. It's not just based on hope anymore. It's based on data. It's based on engineering. It's proof that we can, with enough cleverness, outsmart the virus's best defenses.
Before we sign off, I just want to leave you with a final thought to mull over. We usually think of medicine, and especially vaccines, as being loud alarms, right? We want to wake up the immune system. We want a big reaction. We associate strong medicine with a strong, visible response.
But in this case, the breakthrough came from the exact opposite. It came from silence.
Exactly. The silence of the delivery system, that DNA scaffold, is exactly what allowed the body to finally hear the specific message it needed to survive. And it makes you wonder, what other medical problems might be solved not by adding more noise, not by adding more drugs or more interventions, but by quieting the background, by subtracting the distractions so the body can do what it already knows how to do?
That is a profound question. It seems that sometimes, in biology as in life, less noise really is more signal. By removing the clutter, we finally find the solution.
Thank you so much for listening to this deep dive on the HIV RNA Test Guide podcast. We hope this has been illuminating and give you a real sense of where the science is heading in 2026.
And remember, knowledge is power, but knowing your status is the first most important step. If you need testing or resources, there is a network of over 4,500 labs across the United States that is ready to help you. You are not alone in this.
Stay curious, stay informed, and we will catch you on the next deep dive.
Goodbye.
Bye for now.